Hi guys, my name is Lucas Chen, and today we're going to talk about specification of organizer and organizer pathways in Xenopus.
And because of Mengel's early death, she did not get honored for the Nobel Prize for Physiology or Medicine in 1935. Therefore, Spemann received all the credit for her work. The transplantation experiment was conducted using two new embryos, both in their gastrulous stages. The dorsal lip blastopore from the unpigmented newt species was grafted to the ventral side of the pigmented species. The result was the grafted tissue invaginated and consequently induced a new body axis that gave rise to neural tube and mesodermal structures such as somites. This experiment suggested a new theory that a group of cells could induce and determine development of neighboring cells. Later, scientists further tested the theory of embryonic induction by organizers through the creation of a twinned Xenopus embryo, as we can see on the right side of the slide. The complete removal of embryo.
Induction of the organizer. Nucleus center induces the organizer. How do we get nucleus center? Well, nucleus center is defined by high expression of nodal-related protein. Well, what causes the high nodal expression? First, it's due to cortical rotation. Upon sperm entry, cortical rotation occurs. Because of cortical rotation, beta-catenin stabilizer is able to translocate it to the future dorsal side. And because of these beta-catenin stabilizer, beta-catenin is able to accumulate and be translocated into nucleus, which we call nuclear beta-catenin. And nuclear beta-catenin is able to regulate nodal expression. Second, high mesodermal induction via VEGT and VG1 from the vegetal cells to animal cells will also regulate nodal expression. Right? For instance, VEGT is a transcription factor that regulates the transcription of nodal-related protein. A combination of 1 and 2 leads to nucleus center formation. How does the nucleus center induce the organizer? Well, Wnt signaling pathway signals the future organized cell to express SIMOA. SIMOA and nodal, which expressed by the nucleus center, right, leads to the glucosid expression in the organizer. And glucosid is a marker for it to determine the organizer alongside other proteins secreted by the organizer. Purpose of the organizer. The purpose of the organizer is to pattern the body axis. The organizer is able to pattern dorsal structures along the AP axis, which are chordae synaptomorphies, or chordae shared direct characteristics. So some of the chordae synaptomorphies are notochord, dorsal hollow nerve cord, post-anal tail, and pharyngeal slits. Well, how does the organizer do it? Well, the organizer doesn't tell tissues to become or adapt a certain fate. Instead, induce tissue not to adopt a certain fate. Well, how do they do that? Well, they do that by secreting protein that blocks signaling pathways, which prevent the tissues from becoming or adapting a certain fate. Protein secreted by the organizer. Well, like we mentioned from the last slide, organizer secretes proteins to block a certain pathway, which is able to signal tissue not to adapt a certain fate. And to block the Wnt signaling pathway, the organizer will secrete frisbee. And frisbee structure is similar to that of the Wnt receptor frizzle, but it lacks of the membrane explaining domain, which makes it's a quote-unquote dominant negative, a natural dominant negative protein. It's able to compete with the Wnt, the wild-type Wnt receptor from binding to Wnt, which will affect the signaling of the Wnt. In order to block BMP, the organizer will secrete noggin, and similar to frisbee, it's a natural dominant negative proteins of the normal BMP receptor. And there's some protein that's able to block Wnt, BMP, and nodal, and that is protein called Cerveris. Different regions of the organizer. When we look at the organizer, it's broken down in different regions, and different regions of the organizer express and secretes different proteins. And because of that, it means different regions of the organizer is able to block different pathways, which will lead to specification of different structures. As you can see right here at the orange region, we call this region the deep endomesoderm regions, and a protein that's secreted is Cerberus, and a pathway that's blocked by Cerberus is Wnt, nodal, and BMP, and a tissue that it autonomously contribute is liver, thyroid, and anterior gut. And we move down, right here, the brown region, we call it the head organizer, and the proteins secreted and expressed are noggin, Dirkhoff, cordons, and Gouskoy. And the pathway that's blocked by this region of the organizer is BMP and Wnt, and the tissue that it contribute autonomously are head mesoderm and pharyngeal endoderm. And lastly, the red region we call the trunk tail organizer, and proteins secreted and expressed are noggin, cordon, and phallostatin. And the pathway that's blocked is BMP. And this picture right here is before gastrulation. And after gastrulation, you can see that the movement of the organizer involution, right? The orange regions move up like this and followed by the brown region and the red region, and turns into something look like this, the gastrula. And you can see that the deep endomesoderm region is signaling this region of the tissue to become endoderm mesoderm, and then the head region is signaling the ectoderm here to become the head region, and the trunk region is signaling ectoderm around here to be the trunk and tail region. Here are some equivalent of Spemann organizer on other organisms. So in zebrafish, the organizer analog we call the embryonic shield, and in mouse we call it the Hansen's node or the node, and in chicken we call it the Hansen's node.
The complete removal of embryonic shield results in the lack of notochord and precordial plate. The embryonic shield is necessary for forming notochord, precordial plate, and foreplate, just as Spemann organizer is critical for the development of dorsal structures in newt and xenopus. As shown in the figure on the left, both xenopus and zebrafish have an identical basic body plan. This implies that the embryonic shield in zebrafish is analogous to the xenopus organizer. The figure on the right demonstrates that the transplantation of the shield can induce a complete second axis, including the formation of head, brain, and heart in the zebrafish embryo. The green region is the embryonic shield labeled with fluorescin-linked dextranamine for visualization purposes. The arrow pointing with the label HG, meaning hatching gland, is the transplanted tissue which will give rise to precordial plate and notochord.
As mentioned previously that chick and mouse embryos are similar at neuralist stage, it can be seen here that when the chick embryo is cut off from the yolk and folded together, both embryos look very similar and have similar structures. Now we'll move on to the organizer in chick embryos. During gastrulation, a dark strip starts forming at the posterior end of the embryo. This strip extends from posterior to anterior region. The region is called the primitive streak, which is similar to the nucleus center in xenopus embryos. The anterior-most region of the primitive streak is known as the Hansen's node. Hansen's node is similar to the Spemann organizer in xenopus because if it was transplanted to another region or in other species, it will result in a second axis. Once the Hansen's node has extended to its max length, it starts regressing towards posterior end of the embryo, forming notochord and somites just anterior of the regressing node. Moving on, now we'll discuss the organizer in the mouse embryos. Unlike the chick epiblast, which is flat, the mouse has a cup-shaped epiblast forming the proamniotic cavity, the hollow region in the middle of the embryo. Primitive streak is formed by the movement of epiblast cells. Gastrulation and neurulation stages are very similar to that of chick as the primitive streak induces the Hansen's node.
Signaling that's used to help induce dorsal fates, and then after the mid-blastular transition, the zygotic wave of Wnt signaling will be used to induce ventral fates. And the spamon organizer will actually block ventralization on the dorsal side so that the dorsal side doesn't become ventral, and it does so by secreting anti-Wnt, such as frisbee, which will block X-Wnt8. That's a misconception that a lot of people will have, so just keep that in mind, that Wnt pathway is used for both dorsalization and ventralization. Going on to the third one, the spamon organizer tells the tissues to adopt neural and mesodermal fates. This is a misconception that's a little bit technical. So the spamon organizer, the way it works is that it'll block BMP signaling in the ectoderm and mesodermal tissues. If you take the ectodermal tissue out and let it autonomously develop, it will actually develop into neural tissue on its own. So that shows that the ectoderm will actually be able to develop into the neural tissue without the spamon organizer or BMP signaling. It's just that the spamon organizer is going to be able to block the BMP signaling so that it's basically able to develop what it would normally autonomously be specified to develop as. So same thing with the mesodermal tissue. It'll autonomously develop into what it's specified for. And as for the misconceptions, those are pretty much the major ones that you need to keep in mind.
Here are some study questions. 1. After fertilization, what steps lead to the establishment of the new croup center in the xenopus? 2. Wnt is required to induce the organizer, but the organizer secretes proteins that block Wnt. Explain the contradiction. 3. What would happen if the organizer was removed? Feel free to pause the video at this point to answer the questions. 1. After fertilization, what steps lead to the establishment of the new croup center in the xenopus? Cortical rotation moves the disheveled to the future dorsal side to stabilize nuclear beta-catenin. The overlap between beta-catenin, VEG1, and VEG-T lead to high expression levels of nodal, which defines the new croup center. 2. Wnt is required to induce the organizer, but the organizer secretes proteins that block Wnt. Explain the contradiction.